Cool. Okay, so we are 10, 20 seconds past 10. So let me start sharing the screen at least to make sure things work. It should work, I guess. So, and I'm gonna go to full. Okay, so do you see my screen?
Yes, perfectly.
And my pointer also?
Yes, and your pointer.
Okay. So I'm gonna, let's see if I can make the pointer a little bit larger. It might be better. So accessibility, display, and cursor. Okay, so this should be good.
It's perfect.
Yeah, good, perfect. Okay. Okay. So shall I start, Rosita? Is there anything else we have to wait or?
Sure. Thank you very much.
Okay, sure. I will just start then. So let me move my Zoom control kind of things to a different part of the screen so I can see my slides. Well, good morning, everyone. I hope everybody had a good first 2 days during the workshop. I welcome everybody back to the workshop that we are jointly doing between the two NIH centers, one in Pittsburgh, run by Yvette Bahar, and one is in Urbana-Champaign at Illinois that we run. So I thought before actually we go into the details of what we are going to do, so let's have an overview of the program and what we are trying to accomplish in days 3 and 4, and what are our goals and what we are trying to achieve. And also remind everybody that please feel free. I would be very happy to see a lot of questions during the talk. So feel free to interrupt. I don't know, raise your hand or however you want to signal that you have a question. And I try to remember to invite you to even ask questions during the talk. So as I pointed out, this workshop is a joint effort between two NIH biomedical technology research centers. So one in Urbana, one in Pittsburgh. So we are tasked with kind of developing technologies that are used by the community. So things that not every research group can do by themselves. So we have to provide this technology and make it available to the community so the community can use them. For example, the techniques and the methodology, the software, and the programs that you can download easily and to use for your research purposes. Today and tomorrow, we're going to concentrate on actually more than two, but sort of major areas in molecular simulation, namely, so we introduced actually what is the concept of molecular dynamics, why you need to do molecular dynamics, what are the common applications. Unfortunately, the time is not enough to cover everything, but if we give you actually some idea about the usefulness of the method and how it works in NAMD, the program that we developed in Urbana specifically. Then we also introduce you to the sort of more popular program, VMD. It's a molecular visualization and analysis and modeling tool that you will hear about from John Stone after my talk, and how we use this program to actually as a comprehensive environment to model your system, to initiate your simulation, and to analyze and visualize it later. So, which is a very important component when you're dealing with complicated biological and biomolecular systems. So they're so complex, without visualization, it's almost impossible to do anything meaningful in this domain. So we also cover actually some advanced modeling features or tools that we have developed, again, to make it easy for you to set up the simulation and to initiate a simulation project and to analyze it. So you will hear about those tools as well during these 2 days. So our goal is really to remove the initial barrier for people who want to start using our programs, NAMD and VMD, to make you more comfortable with this software environment. So I really recommend very, very strongly to take the hands-on sessions in the afternoon very seriously I know from experience, I have done it myself when I was young, that I would just go listen to the lectures, and that's the most important part, maybe theoretically speaking. But really our goal here is to get your hands dirty with the program. So you can get started with these programs, use them and run into trouble. We love that. So because then you will ask questions and you will learn and answer you can answer your questions and you can get that problem solved, and you feel much more comfortable with the software. And of course, you can always join the NAMD and VMD mailing list, ask more questions or contact us so we can troubleshoot you later. So I hope that actually you will take advantage of these hands-on sessions, during which we have several people from our group and to help you if you have questions, to specifically talk to you and try to find a solution to your problem. So let me start with a sort of brief introduction of our sort of the MD field and our center, just a few slides, and then we go to briefly talking about MD and why you need to do MD. So MD is a very key tool these days in modern biophysics, and even beyond that. So for any molecular sciences, molecular dynamics have become actually a major tool. So you can see in yellow on this slide, since 1990, when our center kind of started, so you can see the progression of the number of publication in the field of MD or molecular dynamics. So you can see now sort of reaching to, in 2020, we had sort of more than 20,000 papers published applying MD simulations. So now to advertise a little bit about our center, I would like to point out, actually, if you look at the life science application of molecular dynamics, that's shown in blue on this graph, and you can see a sort of pretty large fraction of the simulations are in life sciences about biomolecular systems, proteins, membranes, etc. And from that life science application, if you look at the number of papers that are using the softwares VMD and NAMD in them, you can see that more than half of these publications shown in red are using actually our software. So the softwares that the two software packages that we are going to introduce today, and you hopefully will practice with, are among the most commonly used software packages for molecular simulation and visualization. So here is actually a breakdown of the kind of software the number of users and download that you have. So we have had, since the beginning of the center, we have had more than 500,000 users registered for our software. Just looking at the latest version of VMD 1.9.3, we have had almost 200,000 people registered for that program only. And if you are very, very conservative, just looking at people who have downloaded multiple versions of VMD, indicating that these are active users of the program, we have at least 135,000 users of our software, as indicated, highlighted in this circle here. So, so we have a lot of users who are using our software. And our goal is really to make the software available, to make sure it runs on different machines at different supercomputing centers. It's very performant. It takes advantage of GPU for running simulation or for rendering visualization scenes, et cetera, et cetera. Almost actually all very large simulations and complex systems are done with the software that we are introducing to you today and tomorrow. You can see the number of downloads of the software here. So we have had collectively more than 2.2 million downloads of our software from the beginning, actually, I think it's from 2000. So, and we are projecting that this number continues to grow. You can see the download in 2020 shown here, 177,000, and 2021, we are expecting or projecting it to be even larger. So in the United States alone, we can see that people are using it in all the states across the country, but of course we have also statistics really large number of people are using it from other countries. And for a variety of applications, as you can see, including actually quantum chemistry application. This is a kind of standing out that why people use our software for quantum chemistry applications. And it turns out that because of a specific visualization capabilities that we have to handle very complex and large data sets, that has made our software optimal for quantum chemical applications. So it's a very popular software. And the last thing I make is actually, if you don't believe my numbers, by just looking at the number of people who cite our software, only the software, only 3 papers of our software are sort of used for this statistic that you're looking at. So right now in 2021, this data is about maybe 3 weeks old that I'm showing you. So our software is cited every 1 hour and 8 minutes. So by the time I'm done with my talk, we will have 1 more citation, at least, to our software. So that's really a proof that many, many people are using. Of course, many of them don't publish papers. Many of them publish paper, but maybe they don't cite our software because they forget, or the journal has limitations in the number of references, et cetera, et cetera. But those who publish and who do cite us, we get actually a very large number of citations, reaching 50,000 citations or more as I speak, actually, for our software. I guess the point is really that the software is being used for obvious reasons, and I hope that you get to know these software packages today and tomorrow and start using it for your specific purposes. And let us know if you need more kind of features or capabilities of the software. That's why we are here. So we also have some statistics that these are really, the papers that are citing us have very high impact. You can see the impact factors of the papers that have cited us. Even Nobel Prize winners have cited us. Actually, you can see familiar names like Martin Karplus, Brian Kobilka, Arye Warshel, Hartmut Michel in the Joachim Frank. In the list, the cryo-EM expert is using our software again because of the capabilities that they offer. Okay, so let me now switch gears a little bit and start actually introducing the concept of MD and molecular dynamics. Why do we do MD simulation? Why is it so popular? Why is it so commonly used by molecular people? And I would like to cover a few major applications of the method, especially in the domain of biomolecular simulations, to sort of to introduce the method to those who might not be very familiar with it. I guess everybody in this community now is here because they are fairly familiar with at least one of the applications of MD. And that's why they are here. They want to learn more. But I think an overview of the applications of many different flavors of applications of the method is in order for this workshop. So in general, we do computational work, what we call it computational structural biology, because we would like to think that the methods that we are offering here, the techniques that we are offering here, parallels what is happening in structural biology. So why do you do structural biology? Why do you spend, I don't know, 3 years to solve the crystal structure of a protein or the cryo-EM structure of a protein in complex with DNA? What is the point? The point is you would like to describe that biomolecule at nanoscale, at a very detailed atomistic level, to understand how it functions. And that's exactly the purpose of our business here. We would like to know how proteins function. And of course, the function of a protein, if you freeze a protein, the protein doesn't function because it requires motion, sometimes very small fluctuations, sometimes very large scale conformational changes, as you can see in this example, to accomplish its function. And that's something that is extremely hard to achieve experimentally. The motion of macromolecules, biomolecules, it's very, very difficult. There are biophysical methods, especially these days, that have come along and that can give you some information, NMR, FRET measurements, EPR measurement. They can give you some information about the different degrees of freedom in a moving system, but by far they are way behind what we need to describe in full what happens to a protein and how it moves, how it samples different conformational states during its function. And that's why we would like to look at this. So here you can see, for example, as an example, I'm showing just, this is not about simulation, it's just about the point that structural biology tries to understand molecules at a very atomic level. So you are looking actually as an example of a neurotransmitter transporter with a drug that is used in the clinic for patients. It's used right now with a drug bind in its binding site. So you have all the structural information that you need to maybe improve the structure of the drug, to make it better binder, or anything you want. You see exactly where the drug binds as shown on the left, what amino acids from the protein are engaged with the drug, what happens if you mutate some of those residues, and so on and so forth. So with having these detailed atomistic views, you can actually have a much more rational drug design approach to this problem, for example. Or you can actually find out what is the molecular basis of diseases in these proteins. What happens if one residue that is very important for the particular structural element is mutated, or the charge is reversed, and so on and so forth. So that's why we would like to look at things at nanoscale. So here you're looking at the dynamic version of kind of a protein. We are looking at how ADP and ATP are exchanged in your mitochondria, in all the cells from one side of the membrane to the other side, how it binds. So you can see it's a very dynamic process. You have a very large conformational changes in the protein as the substrate approaches and comes in and binds. And that's something that is very hard to capture experimentally, this whole process. It used to be, you can see the reference actually was a long time ago. We published this out more than 10 years ago, but back then it was one of the sort of longest simulations capturing in full detail with no forces, no biasing, nothing. We captured how a drug, a ligand, an agonist, a substrate, whatever you want to call it, binds a protein. How the protein responds to the presence of this ligand. How we can actually modify the ligand to make it even better binder, a stronger binder, and if you calculate binding affinity in the system, and so on and so forth. So we also made, based on this simulation, we made this conclusion that the whole family of these mitochondrial proteins carry a very large positive kind of field in them, positive potential in them that attracts negatively charged substrates like ADP and ATP and so on and so forth. So really strong biological implications of what we observed during the simulations that we did. This was actually about 100 nanoseconds back then, if I remember. So this is another example of MD simulation that I took from the company D. Shaw. So my good friend Ron Dror did this simulation. It was published again around the same time, right after our initial work. So now in this case, you're looking at the GPCR, G protein coupled receptor, and the structure of this receptor with a ligand bound to it was not even known when this simulation was performed. So you can see the ligand shown in yellowish color. So it spends a lot of time sampling the sort of diffusing in the solution, and then it binds to an initial place in the protein, and then finally it sort of docks and binds to its binding sites. Sort of impressively, this took 5 microseconds of simulation. This was really long simulation back there. So if you look at the final product, and you can see that it stays there for a much longer time, you can, if you look at that structure coming out of the MD simulation, pure MD simulation, and compare it to the structure that came out later, you can see the agreement between the two structures is really impressive. It's really showing and proving that yes, you can actually sample very slow events and very relevant phenomena in biomolecular systems, rather than guessing how it might work and where it might find. So as I said, we would like to also describe the large-scale dynamics of proteins during the function. So here you're seeing an example of a membrane transporter. So this particular protein is coupled to ATP binding. It's called an ABC transporter. So it binds ATP down here in these globular domains that are outside the membrane. And binding ATP and hydrolysis of ATP in these domains is coupled to the motion of the top part of the protein, the helical part that you can see in different colors up here. And that's where the substrate is transported between the two sides of the membrane. So in other words, so this machine couples the ATP energy to transport of the substrate against the chemical gradient or electrochemical gradient across the membrane. But so we can see, of course, in this case, we couldn't do this using equilibrium unbiased simulations because the time scale of the process you are looking at is in a millisecond range. So we cannot go there yet. Hopefully soon we will be there. But we have to induce this process using non-equilibrium methods and biasing protocols that I will cover a little bit tomorrow as a major way of addressing the timescale limitation that we are facing in this business. So here is another example of showing actually how sort of a transporter flips and flops, changing its accessibility between the two sides of the membrane. And again, another point I would like to make is you have access to every atom, every angstrom, and every femtosecond of these simulations, or even shorter if you design it differently. So there is no other methodology that gives you this much spatial and temporal resolution at the same time. So here on the right, for example, we are looking at the number of water molecules that are in the two halves of the protein. You can see the background magenta and brown curves are for the end states. And then the black curve that is fluctuating between the two shows how it sort of goes from one state to another state during the transition that you're observing. So another application of the MD simulation has become actually really modeling of complex systems. You know, it's not trivial. So nature does it on its own. So things come together, self-assemble, they find the right density, they take their time. But if you want to put together models for complex systems and study them, so MD is providing a very nice tool to relax the system, to optimize how different components come together, how the solvent and different environments affect them, and so on and so forth. So I'm showing an example of a chip, sort of kind of application to nanotechnology of this business, in which they were trying to actually cover the chip with antibodies, which is a highly relevant subject these days, and then detect viruses and see if the sample that you're putting in this chip includes any viruses. In order to do that, actually, they were working with this schematic that you see on the left. So what can that tell you instead of just sort of visualizing a concept in a paper? Nothing. There is no prediction coming out of this model. Whereas we tried, and this actually model was put together for over several months with a lot of MD simulation. We are actually putting a sort of complete model. You can see the PEG moieties. Some of them are collapsing on the surface, and some of them are connected to the antibody. We are running this, we can run the simulation now and see how these antibodies interact with each other. Is it too much? Should we change the density? So we can really ask physics-based questions in these models. And just to put this together to get an optimal shape and arrangement of all these components of this huge system, we relied on MD simulations for many, many steps involved in the modeling of this process. So here is another example actually in nanotechnology. So you have a gold particle on the right, which is usually again decorated with some polymer, PEG molecules, or and then you can add on top of that lipid molecules. So you will have at the end a nanoparticle that is covered with lipid molecules. And then you can use that nanoparticle for different purposes. You can see an image of this nanoparticle on the left with a pretty large kind of pretty good resolution, but this doesn't give you any information at the atomistic level. So now we try to actually put together a realistic model representing what we have. So here you see the gold particle. We have decorated it with sulfur atoms that we use to connect PEG molecules initially to the particle. And then we can actually put it in a water box, simulate it even, and then later we can add lipid molecules. You can see actually water molecules already, how they approach the surface of the system, what is the wetting percentage of this molecular system. For example, that was one of the questions that our experimental colleague, Kathy Murphy, was interested. And then you can also decorate them with lipids that we did in the next phase of simulation. Here you see you are adding lipid molecules and see how they aggregate on the surface of these nanoparticles. So this is, as far as I'm concerned, the most realistic representation of the system that they're studying. I don't know why I have this one again, so maybe, okay, so then other examples, you can see here 3 different examples of more biomolecular systems. So here you can see actually a virus capsid and how water, ions, and even drug molecules might use the space between the protein, between 2 proteins in the capsid to get into the capsid or to leave the capsid. So again, I repeat, because these fluctuations happen are natural and they happen on the time scale of the, I'm sorry, at the room temperature that we usually study these things. So not taking into account these fluctuations, you cannot get a realistic representation of the space available between these proteins. By just looking at the space between these 2 proteins, which is unfortunately what many people do, and calculate the, I don't know, the radius profile for a static image. So you get only a very good estimate maybe for how big this orifice can be and how large the molecular species that can use this pathway to get into the cell or into the virus can be. Whereas by providing MD simulation and bringing in thermal fluctuation, you can address this in a much more meaningful way. In fact, just for you that might be interested, if we do a simulation at a very low temperature, nothing happens. We don't have any permeation or any exchange of substrate because things are frozen, as you can guess, and nothing happens. So just looking at a frozen system is not really giving you a complete picture of what is happening. You see an example of binding of a protein to the surface of the membrane. That subject has become a very important and hot topic these days. Many proteins bind membranes in a lipid-dependent manner, so I might cover this also tomorrow. And it's important, we would like to know why there is a need for a specific lipid for this protein to bind and gets activated on the surface of the membrane. Again, one subject that cannot be addressed experimentally in a very detailed manner because we don't have tools that give us what happens to the lipids as the protein approaches the membrane, how they aggregate around the protein, how they bind, where they bind, and so on and so forth. And then on the left, you're seeing actually a very old example of a lipid bilayer. So the goal of all this is, I think I'm summarizing this here. So we would like to calculate ensemble average properties sometimes of the system, sort of microscopic ensembles, and then compare them to macroscopic measurements that we usually do experimentally. So experimentally, you can only measure, one temperature or one pressure, whereas we know at the microscopic level, there are many states with different temperatures, with different pressures, and we have to cover them all, and then provide a molecular basis for function of certain drugs, for example, as I showed in my example, and describe actually the structural changes that are involved in the function of proteins. So again, I'm showing you another example, a very nice example where we coupled the tools that we have with the experimental work. So our colleagues, you can see this came out in Nature a few years ago. Our colleagues had solved the structure of this open channel, shown in green actually in this figure. But then when you look at the protein itself, it doesn't look anything like an open channel. And the question was, how can this structure conduct ions? So we had to put it in the membrane, and you can see the lipids here shown in a very ghost-like kind of representation in this view. And we can see actually lipids, when you place them next to the protein and when you simulate them, you let them relax and find an optimal position around the protein, lipids penetrate all the way to the midpoint of the membrane. And by doing that, so let me play this one more time. So by doing that, they can provide and complete the protein for you. The protein seems to have holes on its side and completely closed on it at the bottom and at the top. But by having this kind of interface between the lipids and the protein, you can complete the pathway required for an ion to go in. So without lipids, it was impossible to explain the function of this open state of this channel. But by placing lipids there and simulating there, now we can actually understand how this protein functions. Oh, don't know why it's going back. Maybe I'm quick. Okay, so another area that so I think molecular simulation has been very powerful in is to describe energy landscapes associated with different molecular events. So I'm showing an example in which we looked at the energy landscape of a full protein in the membrane, which is a very complex and expensive case, but in principle, you can apply the methodology that has matured pretty well in the field to any molecular events. Could be rotation of a side chain, could be binding of a drug. In fact, actually, the methods of free energy that we talk about a little bit tomorrow are used heavily in calculating binding affinities for different drugs, and they're used heavily in the pharmaceutical industry. But we can describe the motion of this, and again, for the motions and exchanges or transitions in a protein structure, and then by doing more sampling of the system, they can describe the energy landscape and even find out what would be the minimum energy path connecting two states. Because experimentally, you can, if you are lucky, you can only capture one or two states or intermediates. There is no way to describe the transition between these intermediates in full with experiments. So we can come in and connect the dots here. So again, I talk about this a little bit tomorrow. So one particular aspect I would like to highlight here is the use of what we call collective variables in this business, because we would like to specifically modulate this particular protein, and we know there are helices or domains that move in a particular manner. So we would like to induce those motions specifically, and then that can only be done by applying these collective variables that allow you to collectively change a particular domain in a protein, translate or rotate or move it in any way you want. So I would like to also highlight the major limitations in this business, as I already pointed out. So we have unparalleled spatial and temporal resolution simultaneously in MD, but we should be always aware of the major limitations, and there is a huge amount of work going into these limitations to address them as much as we can. So the two major limitations of this field, so classical molecular dynamic simulation, one is associated with the time scale. Actually, that applies to any MD simulation, because you have to describe the interactions between between all the particles in the system, in this case atoms. Then you are limited in terms of time scale and the time step that you can take. So because the force and the interaction that you calculate is only valid until the system has moved away from that particular set of positions. As soon as you start moving away from that, then you're dealing with a different set of forces and interaction energies. So therefore, we have to calculate these forces at very, very short time steps. So because of the vibrational rules that we know for atomic systems, we know that we are limited to femtosecond time steps. So this is 10 to the minus 15 seconds. And we have to take a lot of time steps to collect information about, I don't know, nanoscale, nanosecond or microsecond scale motion of the system. So therefore, we are limited by how much calculations we can do. This has become extremely better. So I remember the time when I started this business, we were limited really to nanosecond simulations. And nowadays, we are easily simulating microsecond, tens of microseconds of simulations for very complex systems. So the field has moved on by, I don't know, 2 or 3 orders of magnitude in these 20 years. And I hope that by getting faster and faster computers, the field gets better. But that remains to be a sort of major limitation of MD simulation. You are always interested in longer timescale processes, protein-protein interactions, formation of capsid in viruses. So these are very, very slow processes. And so we can always use faster computers and better algorithms to give you these faster simulations and calculations. Okay, time scale, which also translates in limited sampling, in other words, because you cannot run these simulations long enough to sample your system. The other major limitation in the classical way we describe our molecular system is the force field approximation. So we don't have polarization, we don't have charge transfer, and the force field has to approximate all these properties to give you kind of affordable set of parameters that you can use in your simulation. Otherwise, it becomes too complicated and you have even more issues with the time scale that I described. So there is a kind of trade-off with between the accuracy of your simulation, which relates kind of to the force field approximation that I was describing, and the time scale that you can afford. If you take, I don't know, more approximate approaches and models like coarse graining, for example, then you can run longer simulation. But of course, you lose the accuracy of your simulation. If you take very expensive ways of describing your model, getting better enthalpy, let's say, for example, quantum mechanical calculation, which we can use for our MD simulation these days, then you are even more limited in terms of your time scale. And you can only run, I don't know, maybe half a nanosecond instead of a microsecond of simulation. So there is always a trade-off between these two, and we should be aware and try to select what has been kind of maybe prescribed or tried by many people in the field to find the sweet spot for our problem. But at the same time, we should always be aware of these problems and again, be conscious about them. And if we can address them to a large degree, to some degree, we should do that. Or at least we can discuss them in our papers that we know, we have a lot of polarization in this particular I don't know, ligand, and we are aware that the classical description cannot describe these things, and some of the results might be not very accurate, and so on and so forth. So we should be aware of the shortcomings of our tool and technique in general. So, okay, so now I'm gonna move to sort of other tools that we're gonna cover to some degree in these 2 days. But before I move on, is there any questions? I don't see any hands raised. but maybe I give it a pause.
Hello, Professor.
Yes.
Can you, okay. Yeah, I wanted to ask, so do you see custom hardware being used for molecular simulation work in the future?
Well, I mean, we have it already. So we have a custom hardware. If you look at the machine Anton, so this machine was developed, designed and developed just to do MD simulation, classical MD simulations that we are talking about here. So you have some of the calculations at the level of the hardware implemented. So in that way, they make a very, very fast machine to do the steps involved in MD simulations. But then that machine is only useful for MD simulation, nothing else. So it becomes a single purpose kind of machine. So now, to what degree the future goes after that kind of special purpose machines and specific machines, I don't think it's gonna be the only solution, and it's gonna be the dominant solution in the field, primarily because maybe it's very expensive to develop that. There is only one company, DE Shaw, that has done this, and it cannot be available to many, many users. You're talking about, I don't know, hundreds of thousands of users doing this business. So I don't think that becomes the only solution or the solution in the future, but I can see it has its own advantages and power. And in parallel to what we are doing for sort of general community by softwares like NAMD, Amber, Charm, and GROMACS. So I can see that there is also room for those special purpose machines. In fact, we are using that machine on a regular basis, but only for a limited number of projects. The majority of our projects, and that's the point I'm trying to make, the majority of the projects will be done by the software that we have available to the community for free, and the machines and you can run them on any machine. That's actually one of the missions of our center, to make sure that our software is available on any machine that you can buy, your lab can buy, the cluster that you put together, the new GPU that you want to run on, et cetera, et cetera.
Yeah, I was gonna say the economics of semiconductor manufacturing mean that it's almost inconceivable that they would be widely used special purpose machines. You know, you could imagine, much like D. Shaw's Anton machines, where either a wealthy individual or a well-funded company can develop something like that in a very narrow niche with a small number of machines. The total number on Earth is something like 20. You know, that you can definitely imagine that could be maintained somehow, or a large funding opportunity, sort of like a national supercomputer. You could imagine them building quantity 1 or 2 of such a thing, but the majority of the work in the field will continue to be done using programmable processors, which are, you know, either GPUs or CPU-like hardware.
Completely agree. Thank you, John. That's very important comment. Did we address your question? I don't know who asked the question actually, but I hope we addressed the question. Any more questions?
Yes. Thank you very much.
Okay, great. Okay. Any other questions? I know that I haven't got into the sort of practical details of this business, and maybe it's too early, but I thought maybe I offer the opportunity for any questions that one. Okay. Okay, so let me move on. So
Matt, sorry to interrupt you. So I wanted
Maria.
to make sure that we are in a schedule, and based on John's problems with conflict. So maybe we want to wrap up this part and
I am about to wrap up. Yeah, exactly. I'm about to wrap up my part, and I hope that gives John enough. So we will also cover in these days actually other tools that we developed to help you to set up simulations and put together the elements that you need and to execute the simulation and analyze. One of the tools is called QuickMD. It's actually a tool that is implemented in VMD, and you can use to set up a simulation, run it, and even do some analysis, even interactively during the simulation, I believe. So the others, the last application of the computational tools or MD simulation that I'm going to address is, I think I mentioned this a little bit, is when you want to put together very complex molecular systems. So here you're looking actually a complete model of the HIV virus, HIV capsid that was to put together by a combination of electron microscopy and MD simulations to get actually a nicely relaxed array of many copies of these protein aggregates and assemblies that you can see, and to put this together in a way that integrates experimental information in the modeling approach. So this tool took advantage of another kind of tool that we developed here, another technique that we developed here, MDFF, molecular dynamics flexible fitting, which is heavily used these days and is very relevant to the research of many of you, maybe, in which you can actually use a high resolution structure of a protein and fit it into a low resolution density of another state of the same protein for which we don't have a high resolution structure. So by doing MD simulation, taking into account the shape of the density, you can actually get high resolution structure of the new state of your protein. So if you have any questions about this, please make sure that you reach us and you talk to us about application of this method. Yeah, so this is just another example of the tool, and you can even apply it in different steps to make sure that you get the protein structure that is completely in agreement with your electron density. We use a similar approach actually in terms of fitting ligands also in cryo-EM densities. And we try to make it actually more interactive. So here you can see, instead of doing it by hand, you can actually apply MD simulation on this molecular system, apply force as shown by this arrow interactively with your mouse. You can actually drag the protein and put it into the density rather than trying to put a rigid model into the density. That would be much more difficult. So this is a part of interactive molecular dynamic flexible fitting that we are trying. And at the same time, the system can analyze for you the cross-correlation information and how good you are doing, giving a quantitative metric for the quality of what you're doing. So we also use it for putting together very large systems. I know I'm running out of time. So this is the last example I show, and then I hand it over to John. So we are putting together actually very large systems here. This is something that represents a viral envelope, for example, and we take many steps as shown here, coarse graining the protein, putting it in the system, and then running MD simulation that can give you a good distribution of many proteins that you might have, confine these to a particular density that represent the shape of your sphere, and then convert them back to atomistic model. These are all involving MD simulations actually that you hopefully learn today and tomorrow. And you can put together the best model, the best physical model for these objects that you might be interested. The coronavirus envelope is a good example that we are right now working on to get a very sort of packed and tight model of that is perfect for simulations. Okay, John, so I'm going to stop here. So I'm going to just ask one more time if there are any questions, and then you can continue. Any other questions? I have a few more slides, but I can do them tomorrow.
I just wanted to say that it was extremely interesting subject. Thank you very much.
Thank you. Thank you.
Nice.
Thank you.
So
I forgot actually to add to thank you and your team, Rosita, for making this workshop possible and doing all the hard work in the background. That's really appreciated. Thank you very much.
Sure. Thank you.
So there was one question in the chat asking if all all the different tools are available in VMD 194 and would they run on Linux and Windows? And I think the answer for everything that you saw Emad show is yes. But you'll see some more of these things mentioned in my talk in a minute here. So
Yeah.
any other
Cool.
questions for Emad before he goes? Okay.
No. Okay.
So you
can go.
guys can you see my screen there?
Yes.
Yes.
Let me get PowerPoint to cooperate. That's always nice. There we go. Okay, let me get things fixed. All right. Everybody can hear me okay?
Yeah. You're
All right.
set.
So good morning. My name is John Stone. I'm the main developer of VMD. And this morning, sort of continuing with what you saw in Emad's talk, I'm going to give you an overview of some of the new features in VMD and highlight some of the things that I work on in particular. So as you've heard, VMD gets used for a lot of different types of simulations. It interoperates with all of the different popular molecular dynamics tools first and foremost, but it also supports other types of simulations like quantum chemical calculations, cell scale simulations with lattice cell models, and a variety of other things. Some of the key things that make VMD unique are its diverse support for different types of molecular structure information. So VMD can read cryo-EM density maps, tomograms, all kinds of crystal structures. These are then inputs into VMD, which then using a variety of built-in and external tools can be used to prepare and build your structures to get them ready for running simulations with tools like NAMD. And then of course you can run the simulations in an MD package like NAMD, Amber, LAMMPS, GROMACS, etc., and take those results, those simulation trajectories, read them back into VMD for both visualization and quantitative analyses of various kinds. Some of the things that make VMD unique among other programs are that it runs on large parallel computers. So not only can you run it on your laptop or your desktop computer, but you can run it on large supercomputers using MPI. So if you have analyses that you need to run over tens or thousands of frames, that's something that VMD can help you do. So we've written a lot of different tutorials and training material for VMD. If you go to the VMD website, you can find these listed there. There is actually probably far more material than you'll have time to look at this week. The important part is just to know where you can find these things. And so you can go back and revisit these things at any time when you have the opportunity to look at those things. So the areas that we've been working on developing VMD the most, VMD originally began as a visualization tool. And over time, we've added progressively increased capability for structure preparation and simulation preparation. This means, adapting the program to read a broader range of different structure file formats, building tools to use things like density maps and tomograms to guide modeling, to do things like the molecular dynamics flexible fitting that you just saw Emad give a demo of, where you can fit your all atom crystal structure into a cryo-EM density map, for example. These are, tools that you can now use to build very large complex models where you have to use multiple modalities of structure information in order to build complete structures. We're trying to improve the connectivity of VMD to many other tools. Probably more today than ever before. Our field depends on a large number of tools developed at labs all over the world. And so one of the key things that we try to do is make VMD interoperate with all these other tools developed elsewhere. Of course, VMD has built-in scripting languages and things, so it has many extensions that are built in. So one of the examples is QuickMD, for example, which provides sort of an automated workflow for setting up and analyzing molecular dynamic simulations. One of the things that's of key interest for us going forward is building in mechanisms to enhance the reproducibility of the work that people do. So for example, QuickMD is one of the early tools that does this. It keeps a log of all the different steps that a user follows as they build and simulate a structure. And then by logging all those steps, the process that they've followed can be reproduced by somebody else. They can send that log and it can be automatically read by the program and enable somebody else to either pick up from where that simulation left off or to reproduce the same study on different materials. So just giving you an example, there are a large number of different extensions in VMD. So there's the majority of the program is written in C++, but there's a large fraction of its functionality written in scripting languages. And these are things that were written after the fact. They can be added to the program at any time. In fact, you can write your own extensions to make the program do something that we never envisioned doing. And this is something that we spend a lot of our effort making possible in VMD. And just to give you an example of the many tools that are in VMD, a large fraction of them, maybe about 25-30%, were written by people outside of our laboratory. So all these ones on this list that are underlined in green are tools that were developed by other labs, not by ourselves. So just as examples. So as I said before, VMD is somewhat unique among other visualization and analysis tools in that it runs on parallel computers. This enables it both to do parallel visualization as well as large-scale parallel analysis. Just as a simple example, we did a calculation on a long simulation trajectory doing cross-correlation calculations. This is something that's normally a very compute-intensive process, and VMD exploits things like GPU acceleration to make those calculations faster. But even with GPUs, it still takes a long time. And so just as a sample, we can take something that would have taken 5 years with a single CPU, and we can do that in 19 and a half minutes on a parallel computer with 2,000 nodes and GPUs. And so that's very exciting. That just gives you a flavor for what the kind of things that one can do if you run VMD on a large supercomputer as well as your local clusters. Some of the things that VMD can do that you might not be aware of, in addition to being able to work with atomic structure information, you can do analyses that measure spatial properties of those structures over time. So we have built-in tools, for example, to analyze the probability density over time where, for example, in this case, where ions occupy space in tRNA fragment. And so you can do analyses like this, then you can visualize them. You can show the overlap of these probability density shells with the actual motion of the ions. So just as an example, VMD includes tremendous functionality now, especially for high fidelity rendering. So VMD has for decades supported so-called ray tracing based rendering methods. These are very high fidelity rendering methods. They've been around a long time. And VMD has incorporated ray tracing probably since about 2002. But in the current era, we're able to use high performance GPU accelerators to make these things interactive speed. So you don't have to wait 5 minutes or 40 minutes. You know, some of the movies that Emad showed in his talk, when we first rendered those movies, one of them was an aquaporin channel. I think it would take roughly an hour to render something like that back in the old days. Those things can be done completely interactively today, and they can be done with high quality ambient occlusion lighting. We can run these movie renderings on parallel computers, so you can run huge movie rendering jobs very quickly and get rapid turnaround. And VMD incorporates support for multiple ray tracing engines, including Tachyon, which is the one I wrote, an optics-based version of Tachyon, which I also wrote, and Osprey, which is a ray tracer written by Intel for x86 processors. And so these are all built into VMD, and they can produce these images with very high fidelity. You don't have to export your scene to something like Blender to make these images. I think actually in speaking tomorrow, I believe it is, one of my colleagues, Rafael Bernardi, will show you all kinds of cool things that you can do with that. Just to give you a warm up though, what good is this for you? One of the things that you get out of the advanced rendering features of the program is it takes you less work to make an informative image. So just as an example, on the left, we have a virus capsid that's been chopped open, and you can see the half open capsid is lit with a sort of typical OpenGL based rendering. and it looks rather flat. You know, you can't really see the hemispherical shape of this capsid. You can't see any of the pockets, pores, or cavities in the surface. Whereas if you look on the right, you can immediately, due to the use of high quality ambient occlusion lighting, we can immediately see the three-dimensional structure. We didn't even have to apply things like depth cueing. It's immediately obvious what that structure looks like. You can see very clearly the different pockets, pores, and cavities. And so this is a marked difference. And all you have to do is just use the advanced rendering feature, and that's it. And by turning this on, essentially, it's sort of a button click type operation. You can go from an image like the one on the left to the one on the right. And one of the areas that we're putting a lot of effort in right now is making it possible going forward in future versions of VMD to have the visualizations be done with ray tracing in real time on machines that have the necessary GPU hardware. You'll be able to get images like the one on the right for everything that you do all the time. And so that's very exciting. So giving you some examples of how that gets used in practice. This is that same satellite tobacco mosaic virus. This is one of the first virus structures that was simulated with molecular dynamics in our lab. So just looking at the virus structure, you can see with the use of the sophisticated lighting and things, it helps make the depth relationships of various parts of the structure more visually apparent. You can see pockets and pores in the virus capsid very clearly, and it's easier to just discern the three-dimensional structure in general. This is particularly helpful when you're making figures for a publication, when you don't have the advantage of motion to help viewer see these spatial relationships. The next best thing is to lean on advanced lighting techniques to make these things more apparent. So other examples, ray tracing can produce images with high quality transparency. So these chromatophore images, these are light harvesting structures, LH1, LH2 complexes in the upper left. If you look at the structures down in the bottom left, you see the use of depth of field. This is a focal blur like you'd have in a conventional camera. So you can basically tell the program to focus the image on a particular depth in the image, and then the rest of the image will be out of focus as things are either closer or farther away from the camera. So if you want to use this creatively, you can basically draw the viewer's eye to a particular part of the structure while leaving the rest of the structure there in context. It will be blurred out and it won't be distracting. So if you use this creatively, you'll see some great examples from my colleague Raphael tomorrow. Some other benefits when you're looking at large structures with less sophisticated lighting mechanisms, it's hard, for example, with a large virus capsid. You can see in this HIV capsid, every one of these hexameric subunits in the capsid has a small pore in the center, and you can see that they all show this nice shadowed region. With the use of the ray traced lighting and shading, we can see that shadowed pore very clearly. Without the ray tracing, this would look flat and you wouldn't even realize that there's a hole there. And so this is one of the things we get basically for free by using these more advanced lighting techniques. Another challenge with working with large structures, and this is something we're interested in using VR headsets to assist users with. You can imagine looking in on this hexamer. It's very easy to look at it from the outside. If you want to get a camera view like this one in a crowded space, this is something that's a bit of a challenge with a normal 2D windowed application like VMD. This is an opportunity, particularly for VR headsets, to get your face down in the atomic structure and to use your sort of intuition, moving your head around, to let you look at these things more easily. So this is something we're still interested in working on going forward, is basically adding support for VR headsets for navigating dense scenes like this. Of course, VMD gets used to make lots of journal cover images, things like this. You can do this too. The support for large structures and the built-in rendering features are all you really need to build these kinds of visualizations. The ones you see here were all produced directly in VMD using its built-in renderers and a little bit of Photoshop in the case of this little beam of light that we added here. And the rest, you know, that's just simple stuff to do, really. To give you a summary of some of the new features in the program that we're working on now, one of the big items that's in the new version of VMD is support for Python 3. So VMD now supports both Python 2.x and Python 3. There are a bunch of new features that you'll be hearing about today and tomorrow. My colleague Mariano is going to talk this afternoon. I think we have a little juggling going on in the scheduling today because one of our colleagues is away in New York and couldn't give his talk today. So Mariano is going to talk to you about Molecular, QM tool, and FFTK, some of these other tools. I think you'll hear about QuickMD then also either today or tomorrow. These are plugins that assist with structure building. So they allow you to take either a Z-matrix representation, or you can use a graphical editor to build up a molecule an atom at a time or groups at a time in Molecular. Tools like QuickMD will allow you to then continue and build up the rest of the structure, either starting from crystal structures or adding in solvent, things like this, all the common steps to prepare a simulation. The new versions of VMD add many new routines that use GPU acceleration for things like density map manipulation and processing. I'll give you a few examples of that later in this talk. And of course, as I said, we're putting a lot of energy into high fidelity rendering. And big thing that's been of interest of recent due to the pandemic is the ability to run the program remotely with sort of a desktop-like feel from a remote machine. So we at our lab have been doing this for years using commercial software. And one of the things we're working on now is building in sort of a basic remote visualization feature into VMD so that you can connect to it from a remote system and do basic visualization tasks very easily without having to download massive data sets. Particularly if you're using a laptop remotely, this would let you, for example, run VMD on a supercomputer where the data is and visualize that structure without having to download anything. And so this is an area of ongoing work. So the new version of VMD includes tools for density map segmentation. So these are built into the program. They're accelerated by GPUs. So if you have NVIDIA CUDA capable GPUs, it'll use those to make these things 10 to 20 times faster. The new tools have a graphical interface. There's an MDFF plugin that enables things like rigid body fitting, which used to have to be done with external software. There are built-in analytical tools like interactive histogramming, easy masking to allow you to basically chop parts of your density map off or to focus on a particular substructure on a density map when doing fitting operations. It will launch the density segmentation routines on the GPU. So this graphical interface exposes a bunch of these new features, and by virtue of being interactive and giving you the sort of a guided wizard-like mechanism for interacting with the program, it makes these things a little bit easier to use. Some of these tools, if you have to, I mean, they all have text command interfaces also, but they're just simpler to use when you can type into a GUI and press go. So as an example, here's an example somebody created by using the new masking tool to analyze and label different parts of the structure according to the achieved resolution of those parts of the density map. So they did some analysis on the density map to determine its local resolution, and then those local resolutions are color-coded in the visualization you see here. And that's done in concert with the MDFF tool. We're working on building in clustering analyses into VMD. VMD now contains a number of built-in analytical routines that will allow you to do things like RMSD calculations on GPUs with very high performance. And now we're working on teaching the program to stream large trajectories through the program. There are new ways of doing this that allow us to better exploit GPUs. And this can then allow someone to do an analysis on, say, a 10,000 frame trajectory much more quickly than was ever possible before. And this makes it simpler to do sort of all pairs clustering analysis of various kinds. We're starting with things like RMSD and then working our way towards other types of metrics. And so we're just beginning with those things and making the best use of GPUs, and going forward, we'll be extending this mechanism, and out of core trajectory processing will be used for a variety of other types of trajectory analysis calculations as well. So one of the key things I've been working on recently is the GPU accelerated ray tracing features in the program. I think we have an open mic there somewhere. I hear some other sound there. If somebody can mute themselves. Anyway, one of the new features in the program is support for omnidirectional rendering. So if you want to make a movie for YouTube VR, you can now do that in VMD itself and render omnidirectional movies. The new version of the program now supports so-called RTX acceleration. This allows the state-of-the-art NVIDIA GPUs. This allows, so I think one of our colleagues there still got his mic open.
Yeah, I'm trying to mute him, but I can't. Caleb, can you mute him? Thank you.
Yeah.
Sorry about that.
So the state-of-the-art NVIDIA GPUs have dedicated acceleration for ray tracing. This makes these GPUs significantly faster than the previous models. Just as an example, going from the Volta class GPUs that were released about 3 years ago to the second to most recent generation of GPUs, there was a factor of 8 performance gain. So this is why we're going from sort of batch mode rendering to now looking at using ray tracing interactively all the time to give you these high quality lighting and shading features. This is being supported in hardware by the latest GPUs. And so the newest GPUs actually are newer than these yet, and their aggregate speed up versus something like Volta or its predecessors, Maxwell and Pascal, is in the range of 15 times faster. So just as an example, we recently worked on a planetarium dome show, which shows photosynthetic process in purple bacteria. And this is a very challenging rendering scenario. It involves a photosynthetic organelle, which, if modeled in all atom detail, is about 100 million atoms. Even the parts that we choose to show are on the order of 10 to 20 million atoms, and they're in atomic detail. And then we show many instances of these things and very complex scenes. And we were able to render the entirety of the atomic detail structures in VMD itself with the built-in renderers for this dome show. And we have a special planetarium dome projection mode in the renderers. And so this is this strange, if you look at on the left hand, this circular projection you see here, this is the hemispherical view that a planetarium has in the colored region, or the pictured circular region there. And if you imagine sitting in a planetarium and looking straight up, this is what one of the scenes looks like. There's a photograph in the middle of this slide. You can see an actual planetarium with an audience, and that's what it looks like projected on a large dome. So to give you an idea what that experience is like. Anyway, these things are built into VMD now, and you can go to YouTube and watch this. I highly recommend it. It's a really cool production. And this is part of a bigger planetarium show about the evolution of life on Earth. So very interesting. And this subsection of that movie was rendered entirely in VMD. We've added a lot of support for these VR movies. I think Mariano has got some nice movies that he rendered for a carbon nanotube, for example. I don't know if what the plan is, since one of our speakers had to drop out due to conflict with his travel. I think we will have to see what demos they get to show you. But he was going to show you some bunch of VR movie materials. In case he doesn't, I'll just say that because we've built all these features into VMD, it's becoming possible for you to render VR movies very easily in VMD now. And we expect that over the coming year or two, you will begin to be able to use VMD with the commodity VR headsets very easily. And so right now, we did these things as we have early research prototypes where we did all this stuff. The rendering features are already sort of production features in VMD, and then going forward, supporting them as standard displays is coming soon. So aside from the renderers that are built into VMD, VMD works with other outboard rendering engines. Initially, we've targeted ray tracing engines that do, the types of geometry that are most interesting for molecular scenes. But going forward, we intend to link VMD more closely with high-end commercial rendering tools. So these are all examples of linking VMD to other ray tracing engines through a new cross-platform standard called NARI, which is expected to become a standard later this year. When this standard comes out, this will enable VMD to use a much broader range of renderers and will let you connect it to things like Blender and other tools, presumably even more easily than it does today. Just as an example, NVIDIA worked with the Folding at Home team and linked VMD to the NVIDIA Omniverse RTX renderer to make some very nice movies that use very fancy Hollywood style glassy materials like the ones you see here. So you can make some very cinematic renderings of your proteins if you want to. And again, some of the work that we're doing now to link VMD with these other programs will make that easier going forward. The scale of the systems that VMD can work with has increased dramatically just in the last couple of years. We went from being limited to systems of about 100 million atoms in size now to systems that are in the range of 2 to 4 billion atoms. So we've built large protocell models like the one you see here in the bottom right-hand portion of the screen. This is about 1 billion atoms here, but we've built structures up to about 2 billion atoms. So it's just a question of how much memory your computer has now. The program itself is capable of scaling to this size and doing structure preparation and analysis on this size range. So it's just a matter of having machines that have a sufficient amount of memory. Something of this size takes between 512 gigabytes and a terabyte of memory. But if you have the resources or you have access to a remote supercomputer, you can do these things. So this was actually used recently by Romy Amaro's team and a large group of collaborators to simulate the SARS-CoV-2 variant, so the coronavirus. These are some examples from an interview with Romy and some other researchers in the New York Times. These were renderings done entirely inside of VMD. And then more recently, Romy and some people from our lab and others and a large 28-member team won the Gordon Bell Prize last year. And the salient point about that is that all of these renderings and figures that were shown in our paper were done in VMD due to its ability to work with large size structures. So you too can build and simulate things at this virus scale. It's just a matter of getting access to the computing resources necessary to do these things. But there are, NSF and other institutions, Department of Energy, maintain large supercomputing resources that are available to US researchers. All you have to do is write proposals to get time on these systems. And there are systems not only for large scale computing for the molecular dynamics, but also they have dedicated resources for visualization and analysis. And many of them do support things like remote visualization as well. And with that, I'll finish up and take any questions anybody has.
Thank you so much, John. I just wanted to add that I will share a link to Exceed, which we actually at PSC, Pittsburgh Supercomputing Center, work with. And they can apply for Exceed if they need some computation time. I will share the details of how to apply and get time on supercomputer. Thank you very much. It was very interesting. Thank you.
Yeah. And if, I have to run away here to attend another meeting that's co-scheduled with this event. So I will be missing for about 2 hours. But this afternoon and tomorrow, I think we have a break in the afternoon. And our intent is for some of the developers of the software to be available. So if people have questions about VMD or NAMD or the others, we're happy to try and answer those. Another thing I was going to note is that I think for the hands-on session tomorrow, this is for FFTK in particular. Those of you who are running Windows machines, we may have to post a new version of VMD for you to get around a little bug that we found in one of the recent test builds. And so Mariano and I will make sure to communicate that to all of you so that you're ready for the hands-on session on those parts tomorrow.
Thank you, John. I see one question in the chat. Do you have time to answer?
What's the question? Yeah.
Daniel, do you want to unmute yourself and ask your question? Or I can read it for you if you want. Daniel says, great overview. And are you aware, can you please post a link to low-cost stereo mirror headsets? I had trouble finding one.
So I don't know what, so low cost is subjective. So I think that the cheapest stereo headsets you can get would be available on Amazon under the Google Cardboard naming convention. So Google standardized this sort of low cost stereo HMD mechanism. Originally, they actually made them out of cardboard and simple plastic lenses, and they cost about $10. And a bunch of companies jumped on that design and then made a slightly higher quality version that, maybe they're $20 or something like this, but they can be found on Amazon. And the idea is you take your cell phone and you insert the cell phone into the front half of these glasses, and then you can use your cell phone as the display. So that's something you hopefully already own. So if you have a smartphone that runs Android, for example, you can run this Google applications on there. Things like the YouTube VR will also work with that. So if you have a smartphone and one of these very inexpensive headsets, you can watch the VR movies on YouTube. And let's see what else.
John, those are nice. I've used that. Thank you for that. But no, I wanted the old-fashioned mirror glasses that you could use with your computer screen to look at molecules, and I just haven't seen them.
I think they got killed off by things like the Google Cardboard. Yeah, I think that's the issue, is that much like stereo TVs are now almost impossible to find. I think that's a side effect of the existence of the VR HMDs.
All right, so there's a niche market for us. We'll make some put them out there.
I'm sure that you can still get the mirror glasses, but I haven't seen them in probably 10 years. So I'm not sure where you'd find them.
Okay, that's exactly what I was asking. All right, thanks a lot. Great talk.
Yeah. Other questions? So somebody said, is it possible to run VMD across a fleet of cloud hosts? Yes, that it is. In fact, I've got, so running VMD on the cloud is basically the same as running it on a cluster or a supercomputer. The issue then is, like with anything on the cloud, if you're going to run an MPI job, you have to have the list of host names and things, which is a little bit specific to each kind of cloud. So if you're doing it on Amazon EC2, that's done in a slightly different way than if you're doing it on Microsoft Azure. The mechanics of that are specific to each cloud, but essentially it's like running an MPI job on a cluster. So you can run VMD. In fact, I have a paper where we ran VMD across Amazon EC2 on up to 32 nodes to do large scale parallel movie rendering, things like that, to do parallel analysis. The cloud is just as usable as a supercomputer is once you get the job launching scripts written. So that's really the major issue: job launching and having a shared cloud file system. By default, a lot of these clouds, every machine is isolated. So if you want to have them work collectively, you usually want to set up a shared file system. Any other questions I can help with?
Yes.
Go ahead.
I was going to say that actually some people ask questions about web-based VMD visualization in case they want to kind of share their results with colleagues who might not have VMD installed on their machine. Is there any web-based kind of VMDs? And apparently, Abhishek, I told them we can discuss it maybe later this afternoon if you don't have time, John.
I have time. I still have time right now. It's no problem. I don't have to go until just before noon. So there isn't any built-in feature for that. There are WebGL based viewers. Just they're sort of similar tools to what VMD does. They are purely visualization as far as I know. Because they operate in WebGL, there's a limit to the size of the systems that they can work on. Those things will run in a browser to the extent that they can have access to things like memory resources within a web browser. Like if you look at the PDB has, I think, Molstar and JMol are incorporated into the PDB. Those will work for something like that. I think for us, our view is the remote visualization features of VMD are sort of the angle we're taking to make the powerful visualization features of VMD available in a remote setting on a web browser so that somebody wouldn't have to, that way you can have a collaborative session, for example, and have several people connect to the same VMD. And if one of your collaborators is an experimentalist, they wouldn't have to install anything. You could have a Zoom meeting and be talking or having a FaceTime chat or whatever. And then your VMD session would run and they would be able to watch what you're doing, or they would be able to rotate the molecule and so on, but they wouldn't have to install anything. And the difference there is by doing it via remote visualization, VMD retains all of the high-end capabilities it has, both for working with huge structures that are way beyond what a WebGL tool would be able to do. It would also maintain its ability to do things with the interactive ray tracing. That's something that isn't possible today with the web-based viewers because ray tracing is not part of the WebGL spec. So that's something they just can't do at all.
But it is part of WebGPU though.
Yes, but WebGPU is still an ongoing standard, and what they have standardized in WebGPU is far short of what we can do in hardware.
No, the reason I bring it up is I actually got the program compiling using Emscripten, only using the software renderer though, into WebAssembly. I can talk to you about that later. It actually does work, which is pretty cool.
Okay.
Yeah. So what's your question though?
No, my question was about OpenCL. So is there any OpenCL bindings within VMD right now, or is it just CUDA?
So VMD has OpenCL bindings, but Apple dropped the main place where we were using OpenCL was on Apple hardware. Apple has dropped support for OpenCL, and so that made it less interesting. So we still have them, but the range of hardware that we support going forward, we'll have to see what happens with AMD and Intel. So AMD has made the HIP toolkit CUDA compatible. And I have talked to the AMD people about the idea of supporting all the stuff that we do now in CUDA on their hardware with HIP. That's how we're doing HIP support for NAMD, for example. NAMD supports AMD GPUs that way. Intel has a different approach. They have a different programming model, but their GPUs are probably in practice still 2 to 3 years away for GPUs that matter for people on a desktop. We'll see what they do in the supercomputers and the Argonne Aurora machine first, but I think there's no rush there. It's mostly AMD that you would be interested in using stuff on today. But like I said, this has been complicated by the fact that Apple's dropped support for OpenCL. That by itself makes it far less interesting for us. Any other questions?
Yes, I have one question. So I just posted it. So is there any tool in VMD where I can convert DNA aptamer, which is like a very small DNA sequence, like only like 20 to 25 nucleotide, and convert it in a tertiary structure so that like I can do some kind of like aptamer and protein interaction? Because I couldn't find any software where I can convert like very small DNA to a tertiary structure. So do you have any?
There's nothing built into VMD to do something like that, but you might look at other DNA modeling tools out there to see if there are tools that will do that kind of manipulation.
Oh, okay.
Right. I was going to say that what you're trying to do is a pretty advanced kind of modeling, not much to do with visualization or the basic modeling tools that we have available in VMD for basic simulation. So you're generating a sort of tertiary structure, generating an initial model from scratch based on essentially a sequence is, especially for DNA, we don't have it. For proteins, we have something. So you can specify what is the secondary structure, if it's a helix or if it's a, I don't know, beta sheet, so they can generate small pieces